specifically in Kochi, Kalamassery, that industrial area. And the Sarjana Natural Extracts Limited, ABT, can go plant labels in the industry. So these five are the major players in herbal technology sector. And they employ hundreds of candidates for the various research, R&D and production sector. And this is actually a good place where you can, after your course, you can enter into herbal technology industry sector. So I couldn't go to the details because it's almost past 2. So at around 1500 BC, our Malabar coast was the center of medicinal and aromatic plant production. So this medicinal and aromatic plant root, it was known as the spice root. The spice root. So through the spice root only we were trading medicinal and aromatic plants and various plant products like gums, resins, and plants, food plants, and so on. And our Malabar region, it was at that time it was known as Malabar region. World over in Europe and Arabs, it was known as Malabar region. So this Malabar region was the center of this plant trade around 3000 years before. So we lost that glory during this scientific renaissance, and right now we are regaining that lost glory because we have all the basic facilities to have our own value-added products in this using the diversity of plant material. We have accessibility to all the science and technology tools. We have learned manpower. We have all the basic facilities. And it is only your determination that to have a herbal technology startup of you. So we can regain the lost glory by starting our own enterprises in this herbal technology sector. So thank you very much.
Thank you, sir. Now this session is open for discussion.
Sorry, I couldn't go into the details in the last portion of the session, especially on how to the details on the herbal technology startups because that was included. So I think most of you are from pharmacy backgrounds and also from chemistry backgrounds. So you can also contact me if you have any doubts in this biochemistry or herbal technology. But it is a simple experiment you can do. Just take the methanol extract or acetone extract of curcumin, and this is the THC. You can see curcumin, dimethoxycurcumin, bis-dimethoxycurcumin. It's a simple laboratory experiment. You can differentiate different type of diaryl species. And it's another interesting finding from our laboratory, that is Robert serpentine. Sarpagundi. It's one of the most important plant in traditional medicinal herbs. For what purpose it is used? Sarpagundi for blood pressure or So what is the major compound in Robert serpentina? Reserpine. So this is the compound reserpine. So in addition to Robert serpentina, we have Hookery and Micrantha endemic species in the Western Ghats. So we have carried out detailed investigation into the comparative phytochemical profiling of all the available Robert species in the Western Ghats. So these two are the endemic species, and this last one is the standard reserpine. And here you can see, in the endemic species, it is not reserpine but something else, that is the major compound. So we have isolated this compound and found it as reserpine. So that is the diversity of phytochemicals in plants. It is quite difficult to synthesize such compounds in synthetic laboratory, but the plant contains so much diverse structural features in each species. So these are some common examples of medicinal plants and this Comifora fungi. Then in the local name, Gugulu. Google Sterone. Actually, CDR, Central Drug Research Institute, Lucknow, has developed this Google Sterone as a hypolipid compound. A drug formulation they have developed. Migranorrhea, L-DOPA. There's a central nervous system L-DOPA. That's from Migranorrhea. Then I'm locally Migranorrhea. Real. Nike. And this is Rhytia Tindoria, and the compound is Triptan. Rhytia Tindoria. Copala, Dattapalan. And the importance of this compound is it is the only remedy available till now to treat the skin disease psoriasis. There is no modern medicine to treat psoriasis. There are some ointments and some drugs are there, but those are not effective. But this traditional medicine, that is oil extracted from Rhytia Tindoria, that's the only effective remedy to treat this psoriasis, skin disease psoriasis. And the active compound is dermatophytic compound is Triptan. And then Garcinia cambogia, hydroxycitric acid or HCA. Hydroxycitric acid. Europe chocolate compound hydroxycitric acid. That's an anti-obesity agent. Anti-obesity agent, hydroxycitric acid. So in central Kerala, there are so many small-scale industries. Not just small-scale industries, but large-scale multinational companies are also there extracting this hydroxycitric acid from Garcinia cambogia food grains. And it is being imported as a consequence of chocolates and food products exploiting its anti-obesity property. So several studies have done on these anti-obesity properties, and this has been established as a potential bioactive compound. ആദ്യം കാണുന്ന ചെടി ഏതാണ്? ഏതാണ്? Hypnocarpus pendant. Okay, local name എന്താണ്? പണ്ടൊക്കെ നമ്മുടെ നാട്ടിൽ സാധാരണമായിട്ട് കുറവാണ്, മരോട്ടി. And this is a compound, it's a cyclopentenyl fatty acid. So you can see 5-sided ring and there is a double bond, so it is known as cyclopentenyl. E means one double bond. And cyclopentenyl fatty acid, CFA. So till 1943, this was the only remedy to leprosy. Kushtarogathine against Kushtarogathine treat ചെയ്യാൻ വേണ്ടിയിട്ട് 1943 വരെ ഈ നമ്മുടെ മരോട്ടി എന്ന മരോട്ടി ആണെന്ന് പറഞ്ഞാൽ അറിയത്തില്ല. Foreign nations, especially Europeans, it was known as Chaudmugrao. So till 1943, this was the only remedy to treat leprosy. So in 1943 only sulfonamyl drugs were discovered. So that doesn't mean it is still it is active, but more active synthetic drugs like sulfonamyl drugs were discovered in 1943. So therefrom we are using synthetic drugs. But it is active against this leprobacteria. So it was known as Chaudmugra oil. So just like our rubber plantations, this British Empire has made large plantations of Hypnocarpus throughout their colonies to get enough oil to treat their soldiers. Because at that time this leprosy was a dreadful disease. But right now it is under control. What is this plant? Cerbera odova. It's a common plant in our wetland area. And then the importance, it's known as suicide plant. The seeds from just one root can kill a person. And which is a compound. This is compound Cerbera. And this compound is poisonous at the same time and minute doses it can act as a drug. So right now we are importing this fruits of Cerbera odora to some European countries. I don't know exactly where it is going. Nobody knows. But they are extracting the Cerbera and making it into an effective drug to treat some cardiac issues. So right now one of my research students is working on this plant. How we can convert it. It's a highly poisonous plant. Compound Cerbera. Poisonous compound means that minute doses should be act as drug. So, what are the different possibilities of using this Cerbera as a drug, cardiac drug? Given cardiac arrest. Our presentation is and this is the compound taxol I have mentioned. The most important compound discovered so far. Based on that, NCA assay for around 20 years, they have screened around 12,000 plant species and come out. The major compound they have come out was the sparkling taxol. That is from Taxus bradyfolia. That's a Pacific yew, it's also known as Pacific. So that's not our plant, but this is Taxus baccata, found in our northeastern region. In our last collection to this northeastern region, we could identify this Taxus baccata. Aspirin, it's a common drug. Even blood failure. So it all started from the traditional information that this salic salva plant was being used by doctors to treat gum pain. And based on that information, this salicylic acid was found as the major compound, but being an acid cannot be ingested into the human system. So several modifications were done on the salicylic acid basis. And then ultimately, Bayer company in 1899 come out with this compound aspirin or acetyl salicylic acid with minimum side effects. So this acetyl salicylic acid in only minimum side effects compared to this salicylic acid. So that's the extent of this salix alba and aspirin. Artemisinin is another interesting compound, antimalarial compound, just like quinine. And in 2015, the Nobel Prize for Medicine or Physiology was awarded to Professor Yoyoto from China for her discovery of this artemisinin based on traditional information from Chinese medicinal systems. Just like our Ayurveda, or traditional Chinese medicine is more established than our Ayurveda. So it is in China, it is more acceptable than the modern medicine. So based on the traditional Chinese information, Professor Yoyoto discovered this compound. So this is an example of what we are doing at JN-TBGRI on our planet. This is quite interesting. So we have nearly 260 Garcinia species all over the world, and in India we have nearly 41 species, and in the Western Ghats we have 10 species, and this is the common one, Garcinia gumigata. So in addition to Garcinia gumigata, we have several other species. And this is the economic importance of Garcinia species. Garcinia species can be a source of edible fruits, gambo, coloring material that was isolated. Oils and fats. This is Garcinia. Cocoa butter is isolated from Garcinia indica fruits. So that so much wide is the applications of Garcinia species. So these are the phytochemicals reported from Garcinia species. I am going to go into the details. Particularly biflavonoids, benzophenols, and xanthones. These are the major classes of phytochemicals. So several research activities are going on throughout the world in different laboratories on Garcinia species. And we have worked out on the phytochemistry of all the Garcinia species from vegetables. What are the chemical constituents? What are the pharmacological activities? Genetic constitution of these plants, distribution and diversity of these plants. All these details are available in this book, Diversity of Garcinia Species in the Vegetables. And this is our recent discovery. Garcinia seeds are being considered as a waste product. Its fruit rind is only used as a source of condiment, curry Rindubelia, and also as a source of hydrocyclic acid, HCl or an antioxidant. But the seeds are considered as a waste product, but our investigation revealed that seeds are the source of oils. And that we ഉപയോഗിക്കുന്ന എണ്ണയിൽ 65% നമ്മളിപ്പോൾ ഇമ്പോർട്ട് ചെയ്യുന്നു. അതേസമയം ഈ സീഡ് എന്ന് പറയുന്നത് സെൻട്രൽ കേരള റീജിയനിൽ മാത്രം around 500 tons of Garcinia gamigata seeds we will get annually. So from that 500 tons of Garcinia gamigata seeds we will get nearly 200 tons of oils. So നമ്മൾ ഈ 65% around 75,000 കോടി രൂപയാണ് നമ്മൾ ഒരു വർഷം ഈ എണ്ണ ഇമ്പോർട്ട് ചെയ്യുന്നതിന് വേണ്ടി മാത്രം. At the same time we are wasting these seeds. So these seeds can be used as a source of oils and fats. So it is one of our recent investigation. And another molecule from Garcinia species is in the clinical trial phase or as a non-alcoholic fatty liver disease compound. So this is a typical example of what we are doing in JN-TBGRI, biochemical investigation of hitherto uninvestigated plants from the West Indies region and how we can utilize these plants as source of drugs, aromatic compounds and so on. So it's almost 2 o'clock. 5 minutes. So I'm not going into the details. How medicinal plants can be used as a COVID-19 management. I'm not going into the details because we can discuss better on in another session. So we have seen the diversity of plants. So this is the last portion. The diversity of plants we have seen, the diversity of phytochemicals we have seen, the different medicinal plants and biological compounds we have seen. So now, how we can convert this diversity of information, the rich traditional information as well as the diversity of plants, we have all the basic science and technology tools. Then how we can convert this into a viable small-scale industry or viable products through a small-scale industry. So that's the core of this slide, that is herbal technology startups in medicinal plant sector. So this is the basic idea in starting a herbal technology startup. So it's only an introductory slide. I'm not going into the details. Organizational setup, building plan, process, equipments. For example, if you are going into oils and fats, you have to purchase some oil extraction equipment, like oil extractor, water extractor, and so on. If you are into some herbal products, you have to look into similar equipments for tablet making or extraction equipments and so on. What are the products precisely you are planning? And then waste management, marketing, cost-benefit analysis, license and licenses, timelines, how long you will take. So it will take another session to explain all this, but this is the basics of how we can start the herbal technology startups. And in fact, we are in the process of herbal technology startups in oils and fats sector, in aromatic plants, in herbal drugs. So that we can discuss in detail. So this is the basic concept. First of all, you have to have an idea of organizational setup. It has to be in micro scale or small scale or medium scale or large scale. So you have to register your company as a private limited company or a partnership company or a limited liability partnership company. And then there should be a basic building for laboratory and extraction facilities. The process, what kind of process? In herbal technology, mainly it is extraction process, some formulation process will be there. And then equipments, what are the different kinds of extraction equipments required for this process? Maybe different crushing or powdering, pulverizing machines will be required, tablet making machine will be required, solvent extraction machine. So what are the products precisely you are looking at, like herbal drugs or food additives or what exactly it is? So these are the basic steps you have to. So there are different agencies facilitating this small scale industries in herbal technology. So suppose around 25 lakhs is the cost of herbal technology startups that you have in mind. But you need not take this 25 lakhs. Almost around around 20 lakhs we will get as subsidy from the government. So different government agencies are there, like Department of Industries and Commerce, Kerala Startup Mission, and then KSIDC, KFC. These are different government agencies there to support you in establishing your own smart scale industry. These are the agencies in central government, and there are startup incubators. So what are startup incubators? Means you have an idea that you are confident in developing a particular product, herbal product, whether it is in drug or food additive or aromatic or oils and fats or whatever. But you don't have the basic facilities, you don't have the space, you don't have the instruments. You just approach this startup incubators, and this is one of such startup incubators. Bionest at Kalamasheri. In Trivandrum, the Timer is there in Trivandrum. University of Kerala has a startup facility, incubator facility. So the incubator facility provide all the basic facilities to start your own business concepts. Means they provide enough space, they provide all the instrumentation facilities. For example, this Bionest has this LCMS, HPLC, security, GC, so all the high-end sophisticated instrumentation facilities they have at this incubation centers. So we don't go to purchase all these costly equipment, and they even provide space at a nominal rate. For example, you can start your own business at around 2,500 rupees per month. That's the only rent you have to provide. So that's the importance of startup incubators. These are some of the major herbal technology industries exclusively in Kochi. I'm mentioning only about compounds, alkaloids, and peptides. So what are phenolic compounds? Phenolic compounds means phenol. That's an aromatic ring. I think so. If you have some chemistry background. Coming to the Western Ghats, we have nearly 7,500 plant species. Of these 7,500 plant species, nearly 1,250 are endemic to this region. That means these 1,250 species are found only in our in this Western Ghats region, nowhere else in this world. Out of these 1,250 species, maybe 100 species may have been studied so far for its chemical potentials or potential utilities with regard to its biological aspects. That means nearly 1,200. Anyway, surely about 1,000 species are hitherto uninvestigated for their constituents or potential utilities. JN-TBGRI, Jawaharlal Nehru Tropical Botanic and Research Institute, is one of the premier R&D institutes in the country, engaged in various aspects of plant science research. So it is situated in the foothills of the Western Ghats. We have nearly 300 acres of area. We have nearly 4,500 plant species. Around 45,000 accessions of these 4,500 plant species. So it is one of the largest conservative botanic gardens in Asia itself. So in addition to being a botanic garden, we are also one of the leading R&D institutes in various aspects of plant science research. So as I mentioned, I am coming from the phytochemistry. So let us start with what's phytochemistry. I think you may be familiar with the term phytochemistry. In simple words, it's the chemistry of plants. So we have a diversity of plants around us and what these plants contains. So we are mainly working on that aspect, on that perspective, the chemical compounds in plants. So in simple words, it's the chemistry of plants. So it can be defined as the branch of chemistry that deals with the enormous variety of chemical compounds found in plant kingdom. It also deals with the biosynthesis, metabolism, physiological action, and so on. So it is actually a multidisciplinary subject. So it is related to chemistry, biochemistry, botany, biotechnology. So many other subjects are correlated with this biochemistry. So plants are the starting material for biochemistry. So the most fascinating aspect of phytochemical research is the first stage itself, that is plant collections. See, for a For a synthetic organic chemist, the starting material may be, for example, some petrochemical derived compounds like naphthalene or resin. But for a phytochemist, the starting material is the crude plant material collected from his surroundings. So that's the most fascinating and interesting aspect of phytochemistry. So the diversity of plants. So we can see the whole phytochemical work starts with the diversity of plants around us. So all over we have nearly 4.1 lakhs species of plants on this planet Earth. And coming to India, we have nearly 47,000 plant species, of which nearly 18,000 are in large flowering plants or angiosperms. Coming to the Western Ghats, we have nearly 7,500 plant species. Of these 7,500 plant species, nearly 1,250 are endemic to this region. That means these 1,250 species are found only in this Western Ghats region, nowhere else in this world. Out of these 1,250 species, maybe 100 species may have been studied so far for its chemical constituents or potential utilities with regard to its biological aspects. That means nearly 1,200. Anyway, surely about 1,000 species are hitherto uninvestigated for their constituents or potential utilities. So there are different applications of these plant resources in drugs, cosmetics, perfumes, biofuels. So many diverse applications for these plants. So there lies the importance of plant science research. Even pharmaceutical sciences is also closely associated with this plant science. Actually, all these drugs are derived from. It all started. The history of drugs, you can see, it all started from phytochemicals. So we will see some examples. So this is the development of phytochemistry. So as you know, the primitive man, he was much dependent on plants to meet his primary needs such as food, clothing, shelter, and so on. So as civilization advanced, he learned that some plant materials are poisonous in nature, and some plant materials are medicinal in nature, and thus evolved the traditional herbal medicinal system. And as science and technology advanced, he learned to isolate the active portion from the plant material, like essential oil or gums, resins, and so on. And then he learned to isolate the active compounds from plant material, like for example cinnamon from Cinnamomum camphora, that is one particular cinnamon. And thus evolved phytochemistry as a distinct branch of science. So that is the evolution of phytochemistry. And now we can see the evolution of phytochemicals. So right now we can count nearly 2 lakhs secondary metabolites from literature. So all this diversity of phytochemicals, it all started from this photosynthetic bacteria that appeared around 3 billion years ago. From that tiny creature, now it is up to around 14 into 10 to the power 10 tons of organic matter are being produced annually, leading to around 2 lakhs secondary metabolites. So that is the evolutionary history of phytochemicals. It all started from this photosynthetic bacteria, and then comes the blue-green algae, and then comes the vascular plants. So each secondary metabolite or each structure, as you see here, that bear in its structural diversity an evolutionary history of around 3 billion years. So an extinct plant means that compound evolved in that plant through this 3 billion years of evolutionary history is also extinct. So there lies the importance of conservation of aspects, conserving our diversity of plants. So now we can see how this diversity of plants, phytochemicals are synthesized or manufactured in the plants. So there is biosynthetic pathways, precisely biosynthetic pathways for synthesis or manufacturing of these compounds inside the plants. You see, it is quite difficult to replicate this diversity and complexity of phytochemicals in a synthetic organic chemistry laboratory. Right now we have so much sophisticated instruments and all the facilities, but it is quite impossible to replicate the diversity of compounds as we found from nature. So there lies that the fascinating aspect of plants is that each compound is biosynthesized through some specific biosynthetic pathways. So specifically we can classify the phytochemicals into primary metabolites and secondary metabolites. So primary metabolites, almost all the plant contains carbohydrates, proteins, amino acids, fats. So these are very much essential for the existence of plant life. So you can see cellulose is found on almost all, it is an essential component of every plant, or fat, or protein, or DNA, or RNA. So these are very much essential for the existence of plant life. Without these primary metabolites, plants cannot survive. But these secondary metabolites are derived from primary metabolites through specific biosynthetic pathways. So we can see these are the biosynthetic pathways. For example, phenolics compounds, phenolics are developed from primary metabolites through shikimic acid pathway. Alkaloids are developed through amino acids, and terpenoids through malonic acid pathway. So as I mentioned, we have elaborated nearly 2 lakhs of secondary metabolites of phytochemicals so far. But all these phytochemicals are coming under these 3 classes: phenolics, alkaloids, terpenoids. So basically, this phytochemistry is not that much complex. You have to just have some basic understanding of this biosynthetic pathway, diversity of phytochemicals, and so on. So all these phytochemicals comes under these 3 categories of compounds: phenolic compounds, alkaloids, and then terpenoids. So what are phenolic compounds? Phenolic compounds means phenol. It's an aromatic ring. I think so if you have some chemistry backgrounds, you are chemistry students. So phenolic compounds is nothing but there's an aromatic ring attached to an OH. So that's a phenolic compounds. So if there is such a moiety is there in the structure, you can definitely call it as a phenolic compound. So what is alkaloids? Alkaloids are compounds with at least one nitrogen. The simple definition is most of the alkaloids are poisonous at high doses and drugs at low doses. Most of the drugs being used right now are belonging to the class alkaloids. Most of the traditional drugs are coming under this class of alkaloids. So alkaloids are phytochemicals with at least one nitrogen atom. And then what are terpenoids? You may have heard about this isoprene. Terpenoids are made up of isoprenes, C5H8. So if one isoprene is there, 2 isoprene is there, it is monotropine. Or 3 isoprene is there, is sesquiterpene, that is C15H24. And then diterpene, triterpene, and so on. And this rubber is a polyisoprene. That means this rubber is made up of isoprene units. Several isoprene units are combined together to form this rubber. So these 3 are the basic class of compounds. So these are all about phytochemistry. That means phenolic compounds, alkaloids, and then terpenoids. So these phenolic compounds are derived from primary metabolites, like you can say it starts from malonic acid pathway. So you may have learned about these biosynthetic pathways, basic pathways. Of course, all this starts from photosynthesis, and then from photosynthesis, carbohydrates are formed, and from carbohydrates, malonic acid, pyruvate. That's the basic biosynthetic process. From that malonic acid pathway is biosynthesis of all this. That malonic acid, these phenolics are formed through shikimic acid pathway. And then these amino alkaloids are formed by the combination of different amino acids, and these terpenoids are formed through malonic acid pathway. So this is the nutshell of phytochemistry. And now we can see phytochemical techniques. So we have seen the evolution of phytochemicals. So it all started from photosynthetic bacteria that appeared around 3 billion years before, and gradually comes other developed organisms up to this vascular plants. And now we have nearly 2 lakh secondary metabolites elaborated so far. So all these are biosynthesized through some simple biosynthetic pathways. Mainly we can classify this into primary metabolites and secondary metabolites. So the secondary metabolites we are mainly dealing with in phytochemistry, whereas these primary metabolites such as carbohydrates, fats, or amino acids, lipids, they are mainly dealt by biochemists. So that's the diversity of phytochemicals. And now we can see how do we do this phytochemical research in the laboratory. That's a scheme for phytochemical research. So that's a crude plant material and that's the fine structure of a compound isolated. That's a new compound isolated in our laboratory from that plant. That's ribose cannabis. And how we do that. So that's the phytochemical techniques. For that purpose we use different phytochemical techniques. First of all, selection of plant material. The plant material can be selected based on various aspects like random selection or based on traditional information or high throughput screening. So different aspects of selection are there. So once you have selected a plant for further research, the research may be to develop some modern drugs for some particular ailments such as cancer, anti-cancer herbals, anti-cancer phytochemicals, or anti-diabetic phytochemicals, or some, to develop some perfumes, or some to develop some biofuels. So based on your requirement, you have to select the plant material, the plant part, and then the first step in phytochemistry is extraction. So the good plant material that contains thousands of compounds, so many unwanted things are there. So we are looking for a particular class of compounds like alkaloids or terpenes, or maybe we are looking for all the types of secondary metabolites. So we have to extract these compounds from this plant material. For that we are using different solvents like hexane, chloroform, ethyl acetate, methanol, ethanol. So these are the major solvents used in this. Hexane, that's a low polar solvent. Chloroform, ethyl acetate, acetone. These are medium polar solvents. Methanol, that's a polar, highly polar solvent. Methanol, ethanol, and water. These are considered as highly polar solvents. So if we use hexane, for example, for the extraction purpose, we get low polar compounds. So there is a basic theory for that. Like dissolves like. The basic theory in chemistry. Like dissolves like. So if we use a low polar solvent for the extraction purpose, low polar compounds will get extracted into those solvents. So hexane extract mainly contains low polar compounds like fats or some simple compounds. While these polar solvents like methanol or ethanol or even aqueous water, that extracts polar compounds. Polar compounds means some alkaloids are highly polar, some glycosides, glucose. The glucose contains so many OH groups. So that is basically polar compounds. So glycosides, there are some polar alkaloids. These are all extracted in polar solvents. So that's the details of extraction. So actually, through this session, it is intended to give only an introductory discussion on all these aspects because it will take single session on each this extraction process, separation process, and characterization process. And hope in future we can have detailed discussion on these various phytochemical techniques. So after extracting these compounds in the solvent, like hexane extract or acetone extract or methanol extract, next step is separation. Even that extract contains hundreds of compounds. So we have to separate each compound into pure form. So that separation is an important step in the phytochemical process, and we use chromatography. This is color chromatography and this one is thin layer chromatography, TLC. So through the various chromatographic techniques like as you see, color chromatography, thin layer chromatography, HPLC, HPLC, GC, so many chromatographic techniques are there. So each depends on the nature of compound. So through chromatographic separation, we can separate the compounds into pure form. So once a pure compound is separated from this extract, that may be a white solid or a colorless liquid or an orange crystal. So we don't know what it is. So the final step is structure elucidation. So we have to find out the structure of this compound. So that is the fine structure of the compound. So how do we find that structure? So that is mainly through characterization techniques like spectroscopy. So spectroscopy is the major technique used for characterization purposes. So of that white compound we take different spectra. So what are the major spectroscopic techniques? UV visible, IR, NMR, MS, X-ray crystal. So these five are the major spectroscopic techniques. From UV visible spectra, for example, we get information regarding chromophore groups. Like if there is a C double bond O group, that information we will get from UV visible spectra. From IR spectra or infrared spectra, we get information like functional genes. So just on looking the IR spectra, if there is a peak at 1700, that indicates the presence of what? Carbon, C double bond. And among C double bond, there are different types of carbonyl groups, aldehyde, acidic, so many types. So that change in that 1700, 1650, 1600, that indicates which type of C double bond it is. Similarly, if there is an absorption, if there is a value at around 3200, what indicates in IR spectrum? Around 3200. That's OH2, hydroxyl. So just looking on this IR spectra, if there is a value at 3000, that indicates OH. If there is a value at 1700, that indicates the presence of carbon. So from IR spectra, we get such information on the functional groups of this compound. And what this NMR spectra gives, what are the different types of NMR spectra? H1 NMR, synthetic NMR. From H1 NMR, the number of hydrogen atoms. For example, here, these are all hydrogen atoms. 2 hydrogen atoms are here. So, how many hydrogen atoms are there? And what is its environment? That information you will get from H1 NMR. And then synthetic NMR. How many carbon atoms are there? So, here it is a carbon, 1, 2, 3, 4. So, we can count. So, that much peak should be there in the synthetic NMR. So just by counting the number of peaks on synthetic NMR, we can say how many carbon atoms are there in this compound. And then comes the mass spectra. So what kind of information you are getting from the mass spectra? The molecular weight of the compound plus fragmentation patterns. So that's a vital information, the molecular weight of this compound. And then from the mass fragmentation patterns, you will get an idea of the structural features of this compound. So combining all this information from UV, IR, NMR and MS, we are arranging into this fine structure. So that's a kind of interesting theoretical configurations, that's spectroscopy, while this separation part, that's mainly manual work. You have to do color chromatography continuously in the lab for getting some pure compounds. While after getting this compound, you have to mainly depend on various instrumental techniques. You use IR, NMR, and so this is the basics of phytochemistry. And hope you got some basic idea of phytochemistry. So we have seen the diversity of phytochemicals, the evolution of phytochemicals, and how do we do this phytochemical research. So these are the basic steps in phytochemistry. It's a conventional phytochemistry laboratory, how our lab looked like around 10 years back. So you can see extraction techniques like this is oxalate extraction. It's a Clevenger apparatus, and this is simple water bath, rotary evaporator, color chromatography. So this is how typical phytochemistry lab is for the extraction purposes and separation purposes. Okay, so we have seen the diversity of plants on our planet. We have nearly 4.1 lakhs of plant species on this planet. Particularly in this Western Ghats region, we have nearly 7,500 plant species, of which nearly 1,250 are endemic to this region. And we have seen the diversity of phytochemicals. And now we can see how we can convert this diversity of plants, the diversity of phytochemicals into some value-added products. That's what we are doing in herbal technology. So this herbal technology is now emerged as a promising field for future India, like biotechnology or information technology. So you may have heard, or you must be familiar with these terms, biotechnology or information technology. So this herbal technology is now emerging as a similar importance, with a similar importance like biotechnology or information technology. The global turnover is around 250 billion US dollars of these plant-derived products. So now we are entering into that aspects. The third part of our discussion, that is herbal technology, or how this diversity of plants, the diversity of phytochemicals can be converted into value-added products, and in which respects, which aspects we can use these plants. This diversity of plants, as I mentioned, we have nearly 1,200 plants endemic to our region, endemic to the Western Ghats, and around 1,000 species are advantage. So we don't know whether that can be applied as drugs, aroma compounds, flavoring agents. So these are the applications of these plants. So there is the importance of plant science research, whether it is phytochemistry or it is in pharmaceutical chemistry or pharmacognosy. All these are connected to plants. So this diversity of plants can be used as source of drugs, aroma compounds. Drugs, we are mainly concentrating our session on the application of plants as drugs. And aroma compounds. Most of the perfumes and flavoring substances you are using are based on aromatic plants. See, essential oils are the basic components of most of the perfumes and flavoring agents. And then biofuels. So many sources of biofuels are there. Your fixed oils like coconut oil that can be used as a biofuel. In 1850s, Rudolf Diesel discovered the first diesel engine with the intention of running it on peanut oil. That was the fuel used at that time. So basically, this fixed oil can be used as motor food additives, oils and fats. Oils and fats, that's a very interesting sector of this herbal technology. So we are using these oils and fats on a day-to-day basis. So it's a $250 million US business. And around 200 million tons of oils and fats we are using annually globally. And coming to India, we are using around 20 million tons of oils. And out of this 20 million tons of oils we are using, around 65%, that is around 15 million tons, are imported. So, നിങ്ങൾ ഉപയോഗിക്കുന്ന ഈ എണ്ണകളിൽ 65%, more than half, it's a contradictory to the diversity we are having. Means we are having such a diversity of plant wealth, we are having all the basic facilities, but we are not exploiting our plant diversity. So there is a scope for starting small scale industries in this oils and fats or in this aroma compounds. So now we are mainly going to discuss the scope for herbal technology, startups in herbal technology sectors. So we are now going to focus on herbal drugs. So this is basic concepts of application of medicinal plants and phytochemicals in healthcare sector. This may be a little bit different from what you have learned about modern drugs, but the basics of almost all the modern drugs comes from these medicinal plants. So the good plant material and herbal combinations. So we are using crude plant material and herbal combinations, and we are also using extracts, essential oils, fixed oils. So many plant-derived products we are using, and then pure compounds from plants, and then semi-synthetic derivatives of plants. So these are the applications of medicinal plants and phytochemicals in healthcare sector. So we are using crude plant material as such, and we are using various value-added plant products such as extracts, essential oils, or fixed oils. And we are also using pure compounds isolated from these plants. And then we are using synthetic derivatives of these pure compounds. So this is Syngona species. Cinchona, you may be familiar with the plant cinchona. കേട്ടിട്ടുണ്ടായിരിക്കുമല്ലോ പയണ ചെടിയൊക്കെ. ഇതാണ് അതിൻ്റെ ഇമ്പോർട്ടൻസ് എന്ന് പറയുന്നത്. നമ്മുടെ ചരിത്രത്തിലെ ഏറ്റവും അധികം ആൾക്കാരെ രക്ഷിച്ചിട്ടുള്ള ഒരു കോമ്പൗണ്ട് തന്നെയാണ്. ഏതാണ് That's the most important phytochemical or bioactive compound in the history of humankind that saved the maximum number of human beings. ഏറ്റവും കൂടുതൽ ആൾക്കാരെ മരണത്തിൽ നിന്ന് രക്ഷിച്ചിട്ടുള്ള ഒരു കോമ്പൗണ്ടാണ് എന്തായിരുന്നു റീസെന്റ് ഫിനൈൻ വാസ് ഇൻ യൂസ്. ഈ കോവിഡിന്റെ തുടക്കത്തിൽ ശ്രദ്ധിച്ചു പ്രത്യേകിച്ച് ഫാർമസ്യൂട്ടിക്കൽ സയൻസിൽ പഠിക്കുന്ന സ്റ്റുഡൻസ് അത് ശ്രദ്ധിക്കുന്നു. ഡൊണാൾഡ് ട്രംപ് ഒക്കെ ഫിനൈനുമായിട്ട് ബന്ധപ്പെട്ട് ഹൈഡ്രോക്സിക്ലോറോക്വിൻ എന്ന് പറയുന്ന കോമ്പൗണ്ട് കോവിഡിനെതിരെ ഫലപ്രദമാണെന്ന് പറഞ്ഞിട്ട് തുടക്കത്തിൽ ഇന്ത്യയാണ് ഹൈഡ്രോക്സിക്ലോറോക്വിൻ്റെ ലീഡിംഗ് യൂസ്. So that hydroxychloroquine is derived from quinine. That's a semi-synthetic derivative of quinine. Quinine എന്ന് പറയുന്ന കോമ്പൗണ്ട് സിങ്കോണ ഒഫിഷ്യനാലിസ് എന്ന് പറയുന്ന ചെടിയിൽ നിന്ന് നമ്മൾ ഐസൊലേറ്റ് ചെയ്തിട്ടുണ്ട്. ആ സിങ്കോണ ഒഫിഷ്യനാലിസ് ഐസൊലേറ്റ് ചെയ്തിട്ട് ആദ്യം ആൾക്കാർ യൂസ് ചെയ്തുകൊണ്ടിരുന്നത് സിങ്കോണ ഒഫിഷ്യനാലിസ് എന്ന് പറയുന്ന ആ ചെടിയുടെ ബാർക്കായിരുന്നു. So that was nearly during 1500s. So during that time, that is the 16th century, people are using the bark of Cinchona officinalis as a remedy for intermittent fever. And later on, Europeans found that this Cinchona bark can be a remedy to malaria. And then during 1630, this quinine was isolated from Cinchona officinalis bark by Joseph Pelletier and Cavendish. They have isolated this quinine from Cinchona bark during 1630. And then during 1860, these Cinchona plantations were established all through the world, mainly by European invaders. In India also they have established several plantations of this Cinchona officinalis. You can see the plantations of Cinchona in Ooty, in Darjeeling, there's large plantations of this Cinchona was there. So these are all for isolating quinine. So quinine was the major life-saving drug at that time. So then comes this anti-malarial compounds, artemether and artemether and so on. Artemisinin particularly. That's from the Chinese plant Artemisia annuum. So before that, extract of Cinchona officinalis. So this extract was common during the medieval period as a remedy to intermittent fever. It was known as the tonic water. And then comes the quinine, isolated from this Cinchona bark. This was isolated during 1630. And then comes chloroquine. That's a semi-synthetic derivative from quinine. And then comes hydroxychloroquine, the much controversial compound hydroxychloroquine. So there was this hydroxychloroquine, there was a controversy whether it is efficient against COVID-19 also. But actually this compound is working against the SARS coronavirus, but so many side effects are there. So it is not advisable to give this hydroxychloroquine to patients some kind of complications. Otherwise, this compound is working against SARS coronavirus. So, in fact, more than 50% of all the modern drugs right now we are using are derived from natural products, especially from phytochemicals. So, this is just one example, this quinine. So, it is based on the traditional information that the people from the Andes region. Andes means just like our Western Ghats, it's a mountainous region on the southern America, on the South American continent, the Andes region. So the aboriginals at the Andes region, they were using the bark of this Sycona officinalis as a remedy for intermittent fever. And then based on that information, Europeans isolated this quinine. And from that quinine, later on various much more active compounds like hydroxychloroquine, chloroquine, and several other derivatives of quinines were developed. And now this hydroxychloroquine is an important drug. Quinine also is an important drug. And now there is it is widely used as an anti-malarial compound, and there was some resistance for this quinine, and now there is a comeback of this quinine for anti-malarial treatment. So, as I mentioned, nearly more than 50% of all the modern drugs right now we are using are derived from this natural drugs or phytochemicals, and this is the most common and best cited example, that is quinine from Sycona officinalis. And you can see there are different traditional herbal applications of this traditional herbal medicines like purification, steam inhalation, decoction, alcohol extract, ghee or milk or oil extract. Just as mentioned, there are different phytochemical techniques. Our forefathers were using these different techniques for isolating the active components from these plants. So this decoction means water extract. So alcohol extract is there. Extract on ghee or milk or oil. So that this ghee or milk or oil, these are our non-polar or low polar solvents. So that extract only low polar compounds, where this decoction extracts polar compounds. So even at that time, there was that basic concepts were there, like extraction using polar, low polar solvents or polar solvents. So this is the steps in the development of a herbal drug. So you can see it is not an easy job. The development of a new drug requires around up to $1 billion and around 20 years of research activities. So you can see Taxol or paclitaxel, one of the most important and widely used anti-cancer drug that is derived from Taxus bacata, Taxus bradyfolia. So it all took around 20 years of intense research by the National Cancer Institute of US. So there was a research program from 1960 to 80, spanning around 20 years. So they have screened nearly 12,000 plant species for anti-cancer compounds, and they could come out with only a few compounds, like 5 or 6 compounds. And this Taxol was one of the major discoveries on that research project. So the point is that it is not an easy job, development of a drug. So then we should understand the difference between a drug and a medicine. So up to a drug, it requires so much regulatory aspects are there. So to develop a drug from a crude plant material, first of all you have to find out that there is a particular compound with some biological activity. So that is the discovery of bioactive phytochemicals. So up to that step we can easily do it on laboratories. So just screen for antimicrobial, for anti-inflammatory, for anti-cancer. So many screening assays are there. So you can screen the diversity of plants using different bioassays, and you can come to a particular compound that is showing particular activity. So that's the first step in discovery of drugs. That's the discovery of bioactive phytochemicals. So once you have a compound, the next step is development of a drug. So these two are totally different. So for the development of a drug, you have to carry out these different steps like in vitro studies, in vivo studies, toxicity, stability, pharmacokinetics. So many aspects are there. So the role of pharmaceutical scientists are mainly on this second stage, that is development of a drug from a bioactive compound. See, for example, in our laboratory we have isolated several compounds, and several compounds are there as bioactive phytochemicals, but none have come up to this stage of a drug, because that requires tremendous amount of money, time, efforts, and so on. In our case, for example, one of our compounds are on this toxicity or stability level But the advantage of herbal medicine is that it is not required to come up to this all these regulatory aspects, because this herbal medicine we are using it for centuries. That means its efficacy is established, its safety is established through the application of for centuries we are using this medicinal plants or herbal combinations. So this efficacy and safety are established. So that doesn't require this most of the regulatory portions of this drug development, and that's one of the interesting aspects of herbal medicines compared to modern drugs. So these are some of the medicines developed in India, developed world over, aspirin, artemisinin. So I am not going to the details because it will take much more time. So for the drug development, we need the collaboration of ethnobotany, phytopharmacology, ethnopharmacology, phytopharmacology, so many biochemistry, biotechnology, so many different aspects. It's actually an interdisciplinary effort. So now we can see some common medicinal plants and phytophthalates. Trichopocelanus. Have you heard about this plant? Trichopocelanus. R&D Pancham. So that was one of the major findings from our institute. We have developed this herbal JN. That's a rejuvenator, that's an antibiotic, immunomodulatory drug developed from Trichopocelanus based on a traditional approach. The Carnic tribes, the tribals of the similar forest, the Carnic tribes, they were using this Trichopocelanus as a rejuvenating plant material while going into the interiors of forests. So based on that information, we have looked into the constituents and via activities of this Trichopocelanus and developed this drug gene. Cadranthus rosacea, Shamalari, Chavanagari, but it is one of the most important plants in the history of medicine, plants of bioactive. We traced it in last. These are the compounds from Cadranthus rosacea. Ophiorhiza mangos. This is the compound camptothecin. So you can see this camptothecin is one of the widely used anti-cancer compounds. Its synthetic derivatives like topotecan and pirinotecan, these are also used as anti-cancer compounds. So right now the source of camptothecin is Canthatic acuminata. That's a Chinese plant. From the roots of Canthatic acuminata, it is isolated, and this plant, Ophiorhiza mangos, is a plant common in our area. In most of the paddy fields, you can see this plant, Ophiorhiza mangos, Amalpuri. So this plant contains camptothecin. This is the standard camptothecin, and this is the extract of Ophiorhiza mangos. You can see this extract is almost full of camptothecin. So right now it is being considered as a new natural source of camptothecin. Sciences is also closely associated with this plant science. Actually, all these drugs are derived from so many diverse is the applications for these plants. So there is the importance of plant science research. So they are dealing with the diversity, water issues related to our state. So Rajiv Gandhi Center for Biotechnology, RGCB, this was previously under KCC, now it is under Central